Hi, everyone. I just want to begin by thanking the organizers, Francisco, John, and Brian, for inviting me here. It's really a pleasure to be here. I got to say, I started to have a little twitch in my eye when John was up here talking about February 12th, the deadline for the papers, and I don't usually have that. Well, sometimes I do, but once in a while. And I realized after a little while why I was having such an extreme reaction, and it was because this morning I had an experience. I woke up and I came out, did my showering and whatnot, and came out of the hotel at 7:31, and the bus had left already. So, I know, you need to be a real taskmaster to pull off one of these edited volumes or so on. Anyway, so today I'm going to talk, like Brian said, about genealogy and historical demography. And I just want to note a couple of graduate students, Leandra King and Peter Wilton, who devoted some of their time in graduate school to these topics I'm going to say something about today. Here's a brief overview of what I'm going to talk about. I want to spend some time describing sources of randomness in population genetics, why we put randomness into models and where in the models it exists, and how well those things are justified or not, as the case may be. I want to talk a little bit about diffusion and coalescent theory. So these are two kinds of models, a forward in time one and a backward in time one, and I just want to emphasize that they're basically the same model viewed two different ways. And one thing that they ignore that I'm going to spend almost all of my time talking about is what I'll call the population pedigree. It can also be called the genealogy of the population or organismal pedigree. That's another phrase. And what it means is just the reproductive relationships among individuals in a diploid biparental population. Well, it doesn't need to be well, yes. I'll usually talk about two sexes, and in any case it'll be biparental, but it might be either of those two. I want to show you some simulations of coalescence within population pedigree. So you build a pedigree in a computer and then simulate coalescence times given the pedigree. And I'll show you a number of graphs about that. The basic idea is that coalescent theory on its own ignores the population pedigree, or more accurately, averages over the pedigree. And this is wrong because the pedigree is actually something that exists, right? We don't average over a past event that happened, or shouldn't, conceptually. Then I'm going to show you some new results on the effect of very large families in these pedigrees. So you put in a really big family at some time in the past, and can you see this in a genome-wide pattern of genetic variation? Similarly, I'll talk a little bit about selective sweeps and how they can structure these pedigrees. Before getting started, I just wanted to note some of John's papers that go back a while. This topic of looking at organismal pedigrees or population pedigrees, there aren't a whole lot of papers on it, and a number of them are by John Avise. One of the early ones, the earliest one, I think, is this one by Ball, Nigel, and Avise. And in this paper are a lot of the conclusions that I'll say, and roughly the first half of the talk. They're already in that paper, although I've gone into some more detail in describing some of the coalescent structures within the pedigrees. And then these other two papers are about migration patterns, isolation by distance, but also including pedigrees and asking things. In particular, this paper is about the sampling structure that we would need to implement in order to get a good picture of the demography of a population given a pedigree. And here is the question asked in this paper, is the concordance of gene trees in a habitat, that a linear habitat with migration, when you have these organismal pedigrees. Yep. Okay. So here are the basically three places in which randomness enters into population genetic models. One of them is in the behavior of organisms. The survival and movement of individuals around a habitat, they're finding mates or not finding mates and then having offspring or not. All of this stuff is super important biologically, but in terms of making a model, I don't think we can argue we have really good knowledge of any of this for any given species. Now, that's okay. The models of population genetics end up making very specific assumptions about what individuals do, and they might or might not be applicable to any given species. But one thing that is for certain is that whatever happens, there is a population pedigree, right? This is laid down by the events of reproduction, movement, all of that stuff that happened in the past, and that thing is fixed for a given species. There's just one of them. The second place in which randomness comes into play is in genetic transmission, and this is like a canonical example of a random process, Mendelian inheritance. In this case, I'll be talking about it within these population pedigrees. Another place in which mutation, the randomness enters in is in mutation, and that is just the fact that which base pair and which generation of base pair mutates in really does appear random. And I just add that it happens at a very low rate. All right, so I want to start out by addressing this first question, which is how well do standard model predictions of population genetics account for variation in times to common ancestry among loci within genomes? And the background for this is that all of the genetic variation is due to Mendelian segregation and mutation within the pedigree. And then I'll go into some more detail about very specific models, in particular of very large families and of selective sweeps. And the basic overarching question for these things is what kind of demographic events will structure pedigrees in such a way that we'll see a genome-wide signature on genetic variation. Here's just a slide about the standard, what I'm going to call the standard models of population genetics. An example is the Wright-Fisher model. These make assumptions about a number of things. They typically, at least as a starting point, will even assume a haploid organism, and we happily apply these to diploid organisms all the time. That's generally okay, I would say. They assume a single well-mixed population often, and I'm going to keep that assumption for all of what I talk about today. They assume a constant large population size. I'll use this typical notation of capital N to be the population size. And then they assume selectively neutral genetic variation. It's in, it's in here, in this randomly mating well-mixed part, that the assumptions about the movement of individuals and whom mates with whom are implemented. And in this simple Wright-Fisher model, for example, it's all just completely uniform at random. So based on these kinds of models, the so-called standard models, and I use this term standard to encompass all of population genetic models as you know them, the salient point is that they average over the pedigree. The canonical simple models are the standard models like the Wright-Fisher model. Based on these kinds of models, people have developed diffusion theory to predict gene frequency change forward in time in populations, and also coalescent theory to account for gene genealogies at loci within species. Here's an example that I like of the application of diffusion theory of forward time modeling in population genetics. It's from an old experiment by Bury in 1956 in Drosophila, who maintained 108 laboratory populations of Drosophila for 19 generations. And he started them all at a 50% frequency of a visible mutant and followed them forward in time. Each generation, what he would do is randomly choose 8 males and 8 females, and then propagate the next generation. And what this graph here shows is the histogram of the allele frequencies in these populations, because the N is 16 and they're diploid, there are 32 possible alleles, so the frequency, the count of alleles can go from 0 up to 32 in a given population. All of them start off with 50% frequency in the beginning of the experiment, and then they start to move around. And what the histograms show is that they spread out in the frequency distribution space, and then they start piling up on the boundaries. So some populations will fix for one allele or the other, and that's what these bars represent here. And the fraction of populations that are in those categories increase as this experiment goes on, right? And then there's some also nice stuff about the shape of the distribution in the middle. All of these things can be predicted by the simple Wright-Fisher model. So on the on the right is the prediction of a Wright-Fisher model forward in time under the same conditions, with one exception, and that is that the population size you have to use in your Wright-Fisher model isn't 16 individuals, it's actually 9 individuals. And if you use 16 individuals, then by the end of the experiment, there isn't enough genetic drift, there isn't enough fixation in the populations to account for what was observed in the experiment. So you set the effective population size equal to 9, and you can get a pretty good match, right? Which also matches what's going on in the middle to the extent that you can test it statistically. All right, so that's an example of a forward time analysis in population genetics, and the reason that I bring it up is that the conceptual framework here is that the prediction on the right is a diffusion prediction for a single population starting at frequency 1/2, right? And that prediction says it will spread out and have these probabilities of ending up in all of these different places after 19 generations. And the experiment does that, right? It starts independently 108 times, runs the experiment, and sees whether it matches the predictions of the theory. When we use coalescent theory, we do it in a little different way, right? The most typical, I would say, use of coalescent theory these days is to look at variation among loci in a single genome, right? And here's an example of that for humans. This goes back kind of a ways now, back to the first SNP-map variation paper back in 2001 in Nature. And the observed data are over here. What these are pairwise comparisons of some number of hundreds of base pairs in the human genome, around 12,000 loci randomly distributed across the genome. And what they did is just count the number of differences. So some of the pairs have 0 differences, and that's that number up here. Some have 1 difference, 2 differences, 3 differences, and so on. And, well, this is the table that's in that paper, and the point that they made is that the coalescent model does an okay job of predicting that variation, whereas a simple Poisson model doesn't. And the reason the Poisson model doesn't is that that model would be true if the only variation accounting for all of this variation among loci in the genome, if the only source of that variation was the variation due to mutation. And the coalescent has in it another layer which adds variation in this context, and that is the gene genealogy. In this case, just the coalescence time for the pair of alleles that you sampled at that locus. So here's a picture of that. Just theoretically, you would imagine if you're simulating data like that, what you would do is you would draw a coalescence time for a locus from the distribution up there. That's the coalescent distribution of for the pairwise coalescence time. And then you would put a number of mutations on it, right? And so you'd get some locus 1 here that would have a relatively high, long coalescence time, and a locus 2 that has a shorter coalescence time. And so locus 1, on average, would have more differences between it, and locus 2 would have fewer differences between the 2 sequences. And you do that very, very many times, right? And the trick is here, the thing that I want to emphasize is that what you're really doing when you're doing that, independently applying the coalescent model, is really conceptually the same as in the Bury experiment. You're using a model that has averaged over all of the evolution in a population. And so you're imagining that somehow these different loci in the same genome that you've sampled from come from independent populations. That might or might not be a good idea. It certainly conceptually is incorrect, right? So there are these 2 fundamentally different random experiments, right? The population genetic models we have really apply in this case here. The idea is that we replicate the entire population. That's our prediction, frequency distribution among potential evolution outcomes. And then there's this other one, number 2, which is repetition of the Mendelian inheritance process within a given species on the population pedigree. And that's really how things happen when we apply the coalescent to, when we take data from multiple loci in a given sample, right? 1 is hypothetical for natural populations, and 2 is really where we're at. So I want to account for that. in coalescent theory, bring the population pedigree into it. Here's just a slide emphasizing what happens in the standard models in the context of the Kingman coalescent, the standard neutral coalescent model that people know. And the basic thing that one does in this model in generating a coalescent model is to calculate the coalescence probability, of course. And here's how it works out. It's a little complicated, but anyway, nu I is the number of offspring of haploid individual I. So in a diploid population, there would be 2 N haploid individuals. And this is the number of offspring of the individual I. If I want to know the coalescence probability, I take the probability that a sample is a descendant of individual I, and the second sample, because I'm doing a pairwise calculation right now, and the second sample is another one of the offspring of individual I. That's the product of those 2 probabilities in there. Then I would sum over all of the individuals, haploid individuals in the population here. That gives me the coalescence probability for a randomly sampled pair of samples, given the outcome of reproduction, those nus. And then what Kingman does and what we all do when we do population genetic modeling is we take an average. So that's what these E's here represent. They're the expected value over the process that generates these offspring numbers, right? That's reproduction in the population. And that thing, the outcome of reproduction is, for a single generation, it's the single generation of the population pedigree. So the coalescent and all population genetic standard models average over that in every given generation. And so you get a single probability like this one, P col here, which you then would imagine applies in every single generation in the past, right? Now in truth, it doesn't, right? Here's a little tiny piece of the human population pedigree. I took it from a paper in PLOS One that came out in 2009. It's a extracted part of a much bigger pedigree of Spanish Habsburg King Charles III, who's not shown in this figure. He's 3 generations down here somewhere, and is an interesting individual. He had an inbreeding coefficient of 0.254, and this was the end of the Spanish Habsburg dynasty. It stopped with an imbecile who could not rule or anything, right? So the paper's about that, not to make fun of this guy, but it's a lot of inbreeding in this larger pedigree, and some of it is in this picture I've got here. The point I want to make now is if I take a sample, a single locus copy from Mary here and another copy of the same locus from Philip, then there's a 0 chance in the previous generation. There's no P col here. There's a 0 chance that they have a common ancestor in the previous generation because they don't share parents, right? And there's a 0 chance that they have a common ancestor in generation 2 in the past because they also don't share grandparents. And in this particular case, they have 2 common great-grandparents, and there would be a big probability of coalescence in generation 3 in the past, right? None of this is in the standard models of coalescent theory. So I'm going to show you a bunch of simulations, not much analytical work on this, but simulations. And here's how they work. You generate a pedigree, and here's a depiction of one generation in a pedigree. There are 3 females and 3 males in this little tiny population. And what you do in the computer is just randomly choose a mother and randomly choose a father for each individual, just uniformly at random, which is what is specified by the random mating in a diploid Wright-Fisher model. Do that for some number of generations. If you're being conservative, you want to do it for a very large number of generations, like 10 times n or 100 times n generations. And then sample some present-day individuals, go in and take a gene copy from each of them, or 2 if you want to, and follow those backward in time through the pedigree. Each time they hit an individual, they go with 50% chance to the mother and 50% chance to the father. And we just keep following them through the pedigree until they end up in the same individual, right? If they end up in the same individual, then they'll have a chance to coalesce of 1/2 exactly because of Mendelian inheritance. And they'll have a half chance that they're the two different copies on the two chromosomes in that individual, and then what they have to do is go to the mother and father, right? So we just follow that in simulations, and that's the source of the graphs I'm going to show. I'll start by showing you some coalescence probability distributions through time, and the way these were generated was to simulate 10,000 random pedigrees, then sample one pair of individuals. I'll start with pairwise comparisons. So for each of 10,000 pedigrees, sample 2 individuals, take one gene from each of them, one gene copy from each of them, and simulate 10 to the 8th coalescence times for those 2 individuals on that pedigree. And that will give me a probability of coalescence in each generation in the past for that pedigree and pair. And I do that many, many times, 10,000, and collect those probabilities for each generation into a histogram. Then make a picture like these ones. It's easiest to start over here to sort of interpret this. Here are the probabilities. That 1 there is the probability 1 over 2n that one would calculate in the Wright-Fisher coalescent model using the formula I showed a minute ago. And so what this graph shows is that if I did the exact same simulation experiment that I just described, but instead of having the pedigree in the sample, I would just generate a geometric time with that probability of success equal to 1 over 2n, which happens to be 1 over 2 times 10 to the 5th in this particular example, and collect everything, do everything exactly the same, right? But there's no pedigree here. And so what I would get is a fin of all of these histograms stacked up on one another, sitting right on 1 over 2n, with a little bit of spread because I haven't done an infinite number of replicates in my simulations. And so what actually happens when I do the pedigree version of this is that it doesn't look anything like that, at least not to start. So if I go into generation 1 in the past, almost all of the pedigrees that I have and individual pairs of individuals that I've sampled have a 0 chance of having a coalescent event in the previous generation. And that just means that the 2 individuals I sampled don't share parents. That's all. And in a population of size 10 to the 5th, the chances are that they don't share parents, right? There's actually a teeny little bit of mass out there in the tail of the distribution because once in a while I might get 2 individuals who are half-sibs, for example. But almost always I'll have 0 chance of coalescence until I get back to a time when these 2 individuals share some ancestors, right? Then there's a bunch of complicated stuff, which I've done a little bit of thinking about in terms of describing how that looks. For example, here is the event that they share one ancestor at that generation in the past. And so you get a little hump of probability mass for that event. Anyway, there's a bunch of complicated stuff that happens, and then I get to a situation that looks a lot like the homogeneous one that I have built into the coalescent model or that I can generate in a computer using this sort of scheme that I described. And if I continue further into the past, it will always look like that. So I have this very short period of time where things are very complicated, and then it resolves itself into a relatively homogeneous process with rate of coalescence or probability of coalescence equal to 1 over 2n, just like in the model. So the model's okay as long as I could skip back in time a little bit. Here's a different picture of that same sort of simulation. On the previous graph, you couldn't track individual populations, and here there are 5 different pedigrees, and I'm showing a line for each one of them for the probability of coalescence. And it shows basically the same thing in a different way, that in the early generations, there's a lot of variation in the probability of coalescence for any given pedigree, and then all of them settle down on a probability of coalescence equal to close to 1 over 2n. And in this case, it's a little bit different. It's 1,000 individuals instead of 10 to the 5th, but it's the same basic picture. I'm going to show some other graphs like this, so I wanted to show it to you. And I also want to make the point here that the time scale of that first part, the messy bit before you get back to something that looks like the coalescent, is on the order of log base 2 of the population size, that number of generations. And it makes great sense, right? Because I have 2 parents and 4 grandparents and 8 great-grandparents, and at log base 2 of the population size, that's the time when my ancestors would fill up the population if I let them grow backward in time without any limits. All right. So previously, those are coalescence times, taking the coalescence times as data. So this is a graph that actually generated some fake genetic data, not a whole lot of it, but some anyway. And what it is 10 independent loci, each with a theta of equal to, for n mu equal to 1. And for each of these 10 loci, Tajima's D was calculated. And then it was asked whether I could reject or not the standard model, wondering whether these kinds of strange things that happen in pedigrees would lead to some detectable effect on genetic data. And what the graph shows is for 2 different sample sizes, n equal to 20 and n equal to 100, and for a bunch of different population sizes, down to something very small, in fact, down to 100 individuals, and a sample of 100, all of them, 1 copy from each individual in the population. Using Tajima's D on 10 loci, you can't reject the standard coalescent model, even though you have this pedigree in there. And that's interesting, maybe a teeny bit discouraging or nice. If you like the coalescent, you say the coalescent is very robust. If you're interested in biology, you say, ah! I'm going to say some more encouraging things in a minute for larger samples of loci. All right, so I want to make a couple of conclusions before moving on. And I should move on fast. So even though we have this pedigree, right, which is not accounted for in the models and constrains all the loci in a genome, the standard models work pretty well as long as the population size is reasonably large and also well mixed. Then you can also describe in some more detail these complicated processes that happen in the log base 2 of the population size, number of generations, which are highly inconsistent with the predictions of coalescent theory. A lot of what I've talked about so far is in this paper right here that was in Genetics in 2012. All right, so I'm going to switch over. That's not good. All right, so right there in that big blank spot is, I hope that didn't happen too much. Ooh, look at that. Oh, there we go. There we go. Good, good. All right. There's this paper that came out recently, and it looks at Y chromosomes using microsatellite data and Y chromosomes throughout Asia to study clusters of haplotypes that are highly represented in local populations. And it follows on some other work on a haplotype at the Y chromosome that is thought to descend from Genghis Khan. And in that previous paper, this haplotype was shown to be in populations in Central Asia, total, about 8% of all males carry that one Y chromosome. And what this new paper, which just came out recently, 2015 right there, did was look a little more closely into subpopulations and over a broader geographic range. And so some of the subpopulations that are in Central Asia have a very high frequency of this haplotype. 68% of some individuals in local populations have that haplotype. And so what I did for what I'm going to show you some pictures of in a minute are simulations assuming a fake version of Genghis Khan, where a single male has a huge number of children. The number of children is up to 8% of the population or 68% of the population. And that individual, this maps okay onto Genghis Khan, existed in generation 28 in the past. And then later, human populations are all growing, and there are some estimates of growth of the different haplotypes in this paper. And so after that time, the population grows pretty quickly at 0.3 per generation, a rate of 0.3 per generation. Here are 2 distributions of pairwise coalescence times on a single population pedigree for the case where the children represent 8% of the historical population. This is a picture of the very far left tail of the distribution. So this is a generation 1, 2, 3, 4, up to 40 in the past. Genghis Khan is right here. Fake Genghis Khan is right here. And there's a little spike in coalescence, right? It looks dramatic when I'm looking at that teeny little tail, but it's really a small portion of the distribution. And you can calculate, so for a pair of lineages to coalesce in Genghis Khan, both of them have to be in the 8%, right? And they have to come from Genghis Khan, not the mother, right? Because they had two different mothers in my model most of the time. So they came from Genghis Khan and they coalesced. So 0.08 squared divided by 8, which happens to be about 0.0008, and that's the height of this thing. If you depict that over the entire spectrum of possible coalescence times, you can't see anything. It's just lost in this first bin of coalescence times here, which is 1 over n of time back in the past, very much longer than this. The n here is 10,000. Man, that's unfortunate. When you bump the number of children up to 68% of the population, you can see an effect. So here's the full distribution of coalescence times when the children are 68% of the population. And you can see that in this case, there's a substantial increase on the scale of looking at the whole distribution that you couldn't see anything for 8% in the previous slide. Here you can see a big bump here. But I wasn't sure that would translate into something discernible in fake genetic data instead of taking the coalescence times directly. So what I did is generate genetic data, pairwise differences between these two copies with a theta equal to 0.5. So this is some number of hundreds of base pairs corresponding to some number of hundreds of base pairs in humans. And this is a distribution of the number of differences, kind of like the table I showed before. This fraction of the loci have that number of differences, 0, and then there's 1, 2, 3, 4, 5, and so on. And the two different bars here, this one applies to when there is Genghis Khan with 68% of the population as his children. And this one corresponds to the same demographic model with the growth in the same population size, but without that big family. And you can see that there are differences, but they're actually pretty subtle. And so I could conclude that if I were taking loci with maybe 1,000 or 500 base pair long loci in humans, I would have to sample a whole lot of them before I would be able to pick up a difference like that. This, I should emphasize, is for a sample of 2, right? Just 2 copies of a locus. So what I did next is to look at a very large sample, thinking that there should be some more information there, in particular in the site frequency distribution. So here are 2 kind of complicated graphs I'll go through, but they're for simulations of 10,000 independent loci, each with the theta equal to 0.5. And a sample of 1,000, right? So it's no longer 2, it's 1,000. And this isn't so outlandish at the moment. The now 1,000 genomes paper has a graphs like these ones for site frequencies for the current 1,000 genome sample, which is actually 5,000 chromosomes or 2,500 people. So, we could get those data, in quotes. And the 2 cases I'm depicting here, this is the 8% case where Genghis Khan's children, quote, Genghis Khan's children, make up 8% of the population, and here's when they make up 68% of the population. So there are 2 sets of site frequencies in both pictures. The blue ones are from the toy Genghis Khan example, and the red ones are from the demography without a large family. So the standard neutral demography, but with the growth. So when the 8% when their children are 8% of the population, you really can't tell the difference, at least not visually and with this amount of data. However, when the children make up a really big fraction of the population, 68 in this 68% in this example, you see a very different site frequency spectrum than you do for the case of no big family, the standard neutral case, but with a little bit of growth. And it's different in two places. There's a little hump and a decrease right there. And then there are extra high frequency derived alleles, right? And this is the derived allele frequency spectrum. So I'm plotting it all the way up to 999 copies of the mutant base. And this is the sort of thing that often people attribute to selection, but you can generate these kinds of patterns in populations with migration, which is sort of what this is a little bit like. So in a migration case, there's some chance that you'll have coalescent events structuring a genealogy so that n-1 individuals become separated from one individual. And then I get a bunch of mutations that are in copy n-1 in the sample. And so you can get the same sort of thing here with Genghis Khan structuring the pedigree. This little hump and dip here have to do with the actual event. So I'm getting back to this fake Genghis Khan in the simulation, and then there's a bunch of coalescent events. What it's equivalent to doing is taking my 1,000 sequences and throwing them into two bins. With 68% chance they go in the children of Genghis Khan, and with 1 minus that, the 100% minus that, they go into the not the children of Genghis Khan. Then there are, they have to go to Genghis Khan rather than one of the women, and then they have to go to the two chromosomes of Genghis Khan. So there's this structured sampling that collapses the lineages that are in the family of Genghis Khan onto his two chromosomes, right? And the result of that is that you get a dearth of site frequencies for all of those coalescent events that happen so fast, and then a little bump for the extra ones after that. So this is something that one could detect, right? So if I sent you out to go get 1,000 individuals from that local population that had 68% of Genghis Khan haplotype, maybe you would see this pattern. And I gotta say, one thing is that our demography of Genghis Khan, the fake Genghis Khan toy example here, is pretty unrealistic, right? So I have a single male has 68% of the population and nothing else is going on. And I think what really happened is something more gradual over some longer number of generations that would show a weaker pattern, certainly, than what I've shown here. All right, in the last little bit, I want to talk about selective sweep. So selective sweep is another way that the pedigree could differ from the standard neutral model. So an allele is introduced, sweeps through the population, and somehow structures the pedigree. What this picture here shows you is two pedigrees for a population of 20 or so, and 20 exactly. And the one on the right actually has a sweep in it, and the one on the left doesn't. And it was possible to make a sweep in this small population using a dominant allele and a big selection coefficient of 6. So the individuals who have at least one copy of that allele have, of 5 selection coefficient, but the individuals who have that allele have 6 times as many children as the individuals who don't have that copy. Here are pairwise coalescence distributions of pairwise coalescence times for sweeps. And they're for pretty extreme sweeps. Here's a selection coefficient of 10, and these are actually additive, not dominant, but a selection coefficient of 10, and a sweep that started 50 generations ago. So if the selection coefficient is 10, and you have a sweep going through, it really affects the distribution a lot. But again, it's this tail, the far left tail of the distribution. So it might not be easy to detect that in genetic data. It looks roughly like the pictures for the toy Genghis Khan example I gave for the case where the children make up only 8% of the population. If I reduce the selection coefficient, it's still not even something that is similar to what people estimate for humans. But if I make it equal to 1 instead of 10, I get a very subtle effect. Even for a selective sweep that sweeps through the population in this very picture, there's only a little lump in the tail of the distribution. And here is a little more drilling into those simulations. The previous graphs were for a single realization. These show 10 different realizations of the same process for s equal to 10 and for s equal to 1. So the same two pictures that I had on the previous slide. And these are these coalescent probability trajectories like the one that I showed several slides ago that show you the very high amount of variation that happens within log base 2 of the population size number of generations, and then a very constant probability of coalescence for some number of time, some number of generations, and then here's where the selective sweep happens. And so these are the probabilities of coalescence for the 10 different population pedigrees with a sweep in them. And what these curves here show are the actual trajectory of the allele frequency in those same simulations. So the allele was introduced again in generation 50, and when s is equal to 10, sweeps very quickly up to frequency 1, and has a big effect on the coalescence time, relatively speaking. Relative to this picture where the allele was also introduced in generation 50 and most of them swept to fixation. Several of them did not, but most of them got to fixation in this picture over a longer time frame, right? And that's what accounts for the much subtler effect, is I'm not having a super abrupt event in the pedigree where everyone is related to one individual that existed only some small number of generations ago, maybe 10 in this picture. It's over the whole 50 generations, and there are many more opportunities to escape the sweep in this picture. And the color coding is such that, you know, this one corresponds with that or that. They're color coded to be the same. So you can see that the bump in the coalescence times corresponds roughly with the sweep happening, although the coalescent events happen very early in the sweep, right, when the population size gets really small. So that's just a little extra thing that's of interest to me for theoretical reasons. I don't think this implies that any of these things would be detectable, certainly not in any natural populations that I know about, but it's interesting anyway. All right, conclusions number 2. Historical demographic events like the ones I've described can actually cause genome-wide patterns of deviations from background variation that is described under standard models. However, in large, well-mixed populations, these events need to be pretty extreme, right? So Genghis Khan, toy Genghis Khan, having 68% of the population as his kids. If it's 8%, then it's going to be very, very difficult in that case to detect the signature of the pedigree on the whole genome. Similarly with selective sweeps, if I crank up the selection coefficient way, way high, then I can get an effect on unlinked loci, which is what that was describing. But the selection coefficient does need to be really, really big. And one thing that I want to emphasize is that there's a great payoff when you're looking at relatively subtle signatures like these ones from pedigrees on taking big samples, both numbers of individuals, which I emphasized in making those site frequency distribution pictures, but also numbers of loci, right? Many individuals and many loci. I think now in humans, we're really seeing the payoff of doing that in being able to apply some really sophisticated models to data and get some interesting analytical and statistical results out of them. So with that, I will stop. Thank you very much.